Good afternoon and welcome to your class in NASI 22 or Agricultural Biology. So last time we discussed about photosynthesis and cellular respiration. So please have a look on the video posted. And for this video, we're going to talk about cell reproduction as in preparation for the next unit, which is on genetics. So cell reproduction involves different organisms are divided into two types in the prokaryotes, like in bacteria, and the eukaryotes, like in plants and animals. And there are three schemes for the cell reproduction through binary fission, wherein the DNA replicates and the chromosome segregates and they enter into cytokinesis. While in higher plants, there is what we call mitosis and meiosis cell division, wherein a diploid cell is undergoes different stages of division and it forms into gametes and zygotes. So let's start with prokaryotes, wherein we all know from our previous lessons that prokaryotes don't have nucleus and they have a single chromosome reproduced by binary fission that includes bacteria. So the binary fission is the primary method of reproduction for prokaryotes like the bacteria, and does not require a mate or is asexual reproduction and does not increase the variation in a population. So mostly they are just of the same genetic makeup. So starting with the cell, which contain the chromosome, it will replicate its DNA and then segregate, and cytokinesis occurs. So it's simple as the binary fission simply divides its cells equally. So the steps which are used by bacteria are the cells increase their cell mass slightly and the DNA also, and cell components are then replicated in both sides, and each cell therefore divides into two daughter cells. So that's the binary fission. So it could be both by central growth and apical growth, wherein the growth is within the center. That's the central growth as this divides, and the apical growth is in between on the other sides as well, as you can see. So this is a typical binary fission of bacterial cell. From this, they increase in size through apical and central growths, and then the cell divides. So this is an example of an Escherichia coli dividing by binary fission. So for eukaryotes, they contain a nucleus and membrane-bound organelles, and they are actually asexually reproduced cells by mitosis. So these are the major events in mitosis, wherein the DNA replicates in one cell. So as you can see, from one single strand, it becomes double-stranded, and they enters into mitosis, wherein the whole cell divides. Eventually, they form into two, and eventually they divide into two diploid cells. So mitosis is the division of the cell nucleus and its contents. So it's the phase, and cytokinesis is the division of cell cytoplasm into two identical daughter cells, and chromatin are the combination of the DNA and histone proteins, where diploid means two copies of each chromosome, one paternal and one maternal. So this is the chromosomes in the larger scheme. As you can see, the chromosomes has the nucleus, the inside the cell, and the chromatid, which contain this is called as the telomere, the centromere, and the other side of the telomere, where this contain the DNA, the histones, and the base pairs. So again, the chromosome structure contains First, this is the chromatid. Second is the centromere. The third is the short arm and the long arm, the telomeres. So the telomeres do not code for genes. They keep ends of chromosomes from sticking to each other and prevent loss of genes. So that's the main function. So in the eukaryotes, it contains different cell cycle and stages in growth and division. So we usually in your biology, this has been discussed. So this has the G1 phase, the S phase, the G2 phase, M phase, and the cytokinesis. So it's a cycle. So it should start from somewhere else. So for instance, we start in G1 or the gap 1, and then cells that sees division will goes out here in the cycle. And second is the DNA synthesis phase and the gap 2 phase, and it enters a mitosis, and where it enters again in the gap 1 phase. So the cell cycles, therefore, is start with a if may start in the G1 phase, wherein the cellular contents excluding the chromosomes are duplicated, and these are the interphase. And in this S phase, each of the 46 chromosomes is duplicated by the cell. And then in the gap 2 phase, the cell double checks the duplicated chromosomes for error and making any needed repair. So it enters now to mitosis and eventually into cytokinesis. So it becomes two cells already. So in the G1 phase, this is the first growth stage wherein cell increases in size and prepares to copy its DNA. While in the synthesis phase, the copying of all of DNA's instructions and the chromosomes are duplicated. And in the gap 2 phase, the time between DNA synthesis and mitosis, it's where cell continues growing and needed proteins are produced. And in the M phase or mitotic phase, the cell growth and protein production stop while the cell's energy used to make two daughter cells and called mitosis or karyokinesis or the nuclear division. So in the interphase, this is the resting stage wherein cells carrying on normal activities, chromosomes are invisible, and cell metabolism is occurring and occurs before mitosis. In the interphase, this is what you can see under the microscope. No? In the stages of mitosis is actually divided into four: the prophase, metaphase, anaphase, and telophase or the PMAT. So the cells undergoing mitosis looks like this, as you can see. So these are again, another figure to show you the prophase, when wherein nucleus disappears and chromatin condenses into chromosomes and separation of this introsomes and the formation of mitotic spindle. That's one. So eventually they in the mitotic phase, this the PMAT, the prophase stage. This is the prophase stage of the PMAT. Now, this is how it looks like under the microscope in the prophase stage. Now, in the metaphase stage, it's where the chromosome align in the metaphase plate. So, these are the chromosomes aligning in the metaphase stage of the mitotic phase. And in the metaphase stage is where you can see here. Now, and the next to the prophase and metaphase stage is the anaphase, wherein the chromatid separates already on the opposite poles. So as you can see here, this what looks like the anaphase. So the mitotic spindle actually This is where the aster and the central pair and the mitotic center were the centrosome, and these are the polar microtubule and the kinetochore microtubule. So this what looks like the mitotic spindle. And on the last phase, we're in the telophase, the new nuclear envelope forms and the chromosomes unfold back into chromatin, and the nuclei reappear and the cell continues to elongate. So in the telophase, this is what looks like in the under the microscope. These are in plants and these are in animals. So, and after the PMOT, we have now the cytokinesis. Now it's the daughter cells now divide. So this is what looks like in the cytokinesis, cell plate forming in plant cells. So the life cycle of the cell is actually shown. Mitosis is a cycle with no beginning or end. So as I have said, the mitosis will occur somewhere in the cycle. So up to metaphase. So this is supposed to be an animation, but it doesn't work right now. So let's just proceed on the meiosis gamete production. So we are now in the stage of meiosis, wherein these are similar in many ways to mitosis, but the differences, however, involves two cell division and results in 4 cells with 1/2 the normal genetic information. And when I say diploid, 2N, it's normal amount of genetic material, while haploid N is 1/2 of the genetic material. So in meiosis, this results in the formation of haploid cells. And in humans, these are the ova, egg, and sperm. Ova are produced in the ovaries in the females called oogenesis, and the sperm are produced in the testes of males called spermatogenesis. And the meiosis occurs in 2 phases. We call it meiosis 1 and meiosis 2. So these are how spermatogenesis and oogenesis occurs. This is the sperm formation, and the meiosis 1 and 2, and the egg formation, wherein you can see here the difference between the two. So in meiosis 1, prior to division, this is the S phase, amount of the DNA doubles. So as you can see from the chromosome before DNA replication, this is not double sister chromatids with a centromere, the chromosome after DNA replication. So in metaphase 1, during metaphase 1, the homologous chromosomes line up along the metaphase plate in the equator, and the areas of homologous chromosomes connect at the areas called chiasmata, and genes are exchanged at this connection. So this is what happens in the metaphase 1. So crossing over, segments of homologous chromosomes break and then reattach at similar locations and results in new genetic combination of the offsprings. As you can see, it has both the combinations already. And this is the main advantage of sexual reproduction. This is called the chromosome crossing over. So the chiasmata is this one. So in the anaphase 1, during anaphase 1, each homologous chromosome is pulled toward the opposite sides of the cell. This one is where the chromosome moves on the opposite sides. Unlike mitosis, the centromeres do not break. That's one. Nuclei may or may not reform following division, and cytokinesis may occur or may not occur. And in the meiosis 2, DNA does not double. Chromosomes randomly lined up along the metaphase plate like regular mitosis, and during anaphase 2, this centromere breaks, and each sister chromatid is pulled towards the opposite sides of the cell, and the nuclear reform and cytokinesis usually occurs, although it is often unequal. So these are the overview of the meiosis. The prophase 1, the metaphase 1, the anaphase 1, telophase 1, early telophase, and late telophase. So this is for the prophase 1, and for the second, prophase 2, as you can see, another metaphase 2 and anaphase 2. So instead of two 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 cells chromosomes, so it will, in the telophase 2, it will result in 4. So in meiosis, the reduction, division, genetic recombination occur. The form of cell division by which gametes with half the number of chromosomes are produced, that's it becomes diploid 2N to haploid N. And the meiosis is a sexual reproduction, and two division, meiosis 1 and meiosis 2 occurs. So from 20 in sperm, there are 23 sperm cells with the 23 egg cells, so it is equivalent to 46 zygote. So in meiosis, the sex cells divide to produce gamete, sperm, or egg. And the gametes have half the number of chromosomes and occurs only in gonads or testes or ovaries. In male, spermatogenesis produce sperm, and female, oogenesis, egg or ova. So in spermatogenesis, in human sex cell, the number, the Zn is equivalent to 46, and 20, it is divided into 23 each. So therefore the sperm haploid N is 23 N in the diploid 2N meiosis. In the oogenesis, in the human sex cell, also this also is also equivalent to 23 haploid, and but then the rest becomes polar bodies which die eventually. So interphase 1 is similar to also to mitosis interphase, yet the chromosome DNA replicate in the S phase. Each duplicated chromosome consists of two identical sister chromatids attached at the centromeres, and centriole pairs also replicate at the same time. And then the nucleus and nucleolus are visible in the chromatin or cell membrane. This is the nucleus and the nucleolus. So the meiosis 1 have 4 phases. The cell division that reduces the chromosome number by 1/2. So the first phase is prophase 1, and this is what prophase 1 is, followed by the metaphase and anaphase and telophase 1. So in the prophase 1, the longest and most complex phase, where in 90% occurs, chromosomes condense, then synapses occurs, become homologous chromosomes come together to form tetrad. And tetrad is 2 chromosomes or 4 chromatids, sister and non-sister chromatids. So the non-sister chromatids are homologs, contain DNA that codes for the same genes but different versions of those genes. These are the homologous regions code for the same gene, and genes occur at the same loci. So this is what look like. Now these are the sister chromatids, exact replicates, but homologous chromosomes are not. So this is how it looks like inside the microscopic structure. So this is the prophase 1 synapses. Again, the homologous chromosomes. These are the sister chromatids, which doubles or tet, and sister chromatids. So the homologous chromosomes are pair of chromosomes with the maternal and paternal similar in shape and size already. So this is the loci. Homologous pairs or tetrads carry genes controlling the same inherited traits, and each locus position of a gene is in the same position on homologs, and humans have 23 pairs of homologous chromosomes, the first 22 pairs of the autosomes, and the last pair of sex chromosomes. So the 23rd pair is actually the sex chromosomes. So this is the homologous chromosomes, the eye color locus and the hair color locus in the paternal side and in the maternal side. So crossing over again may occur between non-sister chromatids that sites called chiasmata, and crossing over involves segments of non-sister chromatids break and reattach to the other chromatid. The chiasmata or chiasma are where the chromosomes touch each other and exchange genes called crossing over. And this causes the genetic recombination. So again, this shows you the genetic recombination wherein the non-sister chromatids are combined. This is the chiasmata, the site of crossing over, and then it, as you can see, it interchange and become new variant. So again, the synapses pairing of homologous chromosome from the paternal and maternal side, they cross over and split into two, but then with the paternal and maternal chromosomes. So in the sex chromosome, the X chromosome of the female becomes, and these are also the X and Y chromosome for the male. So definitely if the new baby or the new cell is a female, it has both the X double X chromosome of the maternal, while for if it's a male, it has both the X and Y chromosome from the maternal and the paternal. So it shows again here. So in the prophase 1, the nucleus and nucleus disappear, the spindle forms, the chromosomes coil and synapses occur or pairing, and tetrads form and crossing over occurs. So this is the tetrad. In the metaphase 1, it is the shortest phase. The tetrads align in the equator. An independent assortment occurs wherein chromosomes separate randomly, causing genetic recombination again. So for instance, we have the A and the B. So this will combine to make AB, AB, big ABs and capital A, B, and small AB, and again recombine into capital A and B and small a and capital B. So in the metaphase 1, the homologs line up at the equator, or this is known as the metaphase. So from 2n is equivalent already to 4, then 1n is equivalent to 2, therefore 2 squared is equal to 4 combinations. So in terms of independent assortment, how many different combination of sperm could a human male produce? So again, from 2n, the human chromosome is equivalent to 46 because each parental has 23 chromosomes. So therefore, there could be 2 to the 23rd is 8 million combinations. So it's so huge. Now in the anaphase 1, the homologous chromosomes separate and move towards the poles, while the sister chromatids remain attached at the center of the centromeres. So this is the homolog separation in the anaphase 1. In the telophase 1, each pole now has haploid, one end set of chromosomes, and in the cytokinesis occurs, and the 2 haploid daughter cells are already formed. In the telophase 1, cytokinesis occur until eventually they separate, just like this. Now we are now in the meiosis 2. There is no interphase 2 or very short only and no DNA replication. And meiosis 2 is similar to mitosis in this side. In the prophase 2 is also same in prophase in mitosis, wherein the nucleus and nucleus disappear and chromosomes condense and spindle forms. And in the metaphase 2, same as also as metaphase in the mitosis, wherein the chromosomes not line up. No. In the anaphase 2, same as anaphase in mitosis, wherein the sister chromatids separate. In the telophase 2, the same as telophase in mitosis, the nuclei and nuclear reform, spindle disappears, cytogenesis occur, and the 4 haploid daughter cells are produced called gametes, which is in either eggs or sperm. So one in sperm cell fertilizes one in. So this what occurs in telophase 2 until they eventually break up into 2 daughter cells. So the variation, also known as genetic recombination, is very important to population as the raw material for natural selection. All organisms are not alike. So each of us is unique. And the strongest, most fit survive to reproduce and pass on the traits. So usually the greatest traits or the most or the stronger or the most favorable traits is the one that is being passed on. So what are the 3 sources of genetic recombination or variation? So we have crossing over or prophase 1 and independent assortment in metaphase 1 and then the random fertilization. So a cell containing 20 chromosomes diploid at the beginning of meiosis would, at its completion, produce cells containing how many chromosomes? So 10 chromosomes haploid or 1N because it's diploid, 20 and 10. So a karyotype is an organized picture of the chromosomes of human arranged in pairs by size from largest to smallest, as you can see. These are karyotype with pairs 1 to 22, from 1 to 22, called autosomes, and the last pair are the sex chromosomes. So the male is the XY, while the female is XX. So we have the so-called Down syndrome or trisomy 21, wherein this occurs in female, wherein they have 47 plus 21. So in fertilization, the fusion of a sperm and egg to form a zygote is a fertilized egg, wherein the 23 egg with a 23 sperm will double to N 46 zygote. So a cell containing 40 chromatids at the beginning of meiosis would at its completion produce cells containing how many chromosomes? So again, 10 chromosomes. Because it's still the same, 20 chromosomes. So thank you very much for watching this video. If you have questions, you just comment on the posted video on cell reproduction, and hopefully I'm going to see you again on the next video. Thank you for watching.